Okay, thank you. And now it is time for Vesa to present the scale-up and commercialization of microbe-based solutions.
Yes, good early afternoon, everyone. So we are now moving towards scale-up and commercialization. And in the SIMBA project, we had a specific work package to that end, and it was titled Processes, Products and Services for Market Uptake. And this is a work package where practically all SIMBA research topics come together when they reach the stage of scaling up and aim to market entry. And unlike the other work packages presented so far, this work package doesn't have a so-called own research topic, but it continues with the work done in other work packages. And this slide is just to show that the majority, 14 out of 23 project partners participated in work package 6 to do work in scaling up and aiming towards commercialization. And in addition to the main objective of this work package, it had specific objectives related to the other work packages. The first one is validation of microbiome production at pilot scale, and the microbes were soil microbes from work package 2, marine microbes from work package 3, and microbes for feed and food processing from work package 4. And the second specific objective was establishing a demonstration of cryopreservation technique for microbiome long-time storage. Third one was introduction of rapid test for identification of microbes from various sources. And the next one, scale up and industrial validation of the production of selected product groups. And the groups were crops, microalgae, and fermented foods. And the final specific objective was development of professional marketing plans for microbial products for agricultural applications. As a scale-up and market-oriented work package, this work package 6 does not have its own topic, so it focuses on further development of the work, those innovations done in other work packages. And actually, each of these tasks described in the chart show a specific research topic which has been carried out in other work packages in the SIMBA project. And now in the next few slides, I'll just briefly go through the key results obtained in work package 6 and what their specific impact is. In work package 1, in task 6 was 1, which was titled Validation of Microbe Production Processes at Pilot Scale. There were 2 key results. The first one was dried or frozen microbial biomass for food processing. And it was exemplified by microbial starters, which were produced and processed for use in the manufacture of fermented foodstuffs. And these available microbial starters were suitable for long-term use, and they will advance the use of microbes in food production and processing. And the second key result was dried or frozen microbial biomass for promoting plant growth. And this was exemplified by microbial starters, which were produced and processed for use as plant growth promoters. And these different microbial consortia obtained with sustainable activities can be used to amend soil to enhance plant growth. And in the task 6.2, which is titled Cryopreservation of Complex Microbiomes for Long-Term Usage and Stability, the key result is cryopreservation technologies developed and applied for long-term storage of single microbes and complex microbiomes. And this was achieved by microbes subjected to cryopreservation for long-term storage, and the microbes were plant microbes, specific fruit microbes, and complex marine microbiomes. This gives possibility for long-term storage, which renders use of microbes feasible in a wide variety of different kind of applications. And in task 6.3, development of rapid tests for identification of microbes, the key result was characterization and identification of microbes in various environments. And this was achieved by development of biochemical and genetic markers as diagnostic features to identify single microbes, and developing rapid tests based on DNA analysis for quantification of specific microbial strains or complex microbiomes. And this gives possibility to identify single microbes and monitor the succession of complex microbiota in industrial processes. For example, it provides tools for better process control. And in task 6.4, scale up and industrial validation of the production of crops, microalgae and fermented food, there were in total 3 specific key results. The first one was scaled up production of microalgae in which enhanced capacity for a production of algal biomass and food and feed will increase the resilience and sustainability of food systems. And the second one is a scalable process for the production of fermented food. In this biomass for fermented food, upgraded and validated to industrial pilot scale, will broaden the variety of vegetable foods, and this is especially important for vegans and also those people who want to increase the share of plants in their diet. And the third key result is a virtual database of the project's microbe culture collection, in which draft annotated genomes to be submitted to publicly accessible databases will add data to the database of microbes and microbiomes of the project. And finally, in task 6.4, professional marketing for microbiological products for agriculture, the key results was a strategy to introduce microbial products for the benefit of key stakeholders. And the stakeholders in this case are most likely farmers. And the main purpose is that the stakeholder farmers will be sensitized of new products and services to increase their competitiveness on the market. This is quite an overcrowded flowchart, but just to be brief, it shows the contribution of these specific results obtained in Work Package 6 to the SIMBA's overarching objectives. The overarching objective improve the overall knowledge of microbiomes from land and sea to what the market needs was supported by key results related to microbial growth and long-term storage. And the second one, get a better understanding of microbial structures and functions related to land and sea and related to food chains. Also that was supported by the result development of rapid tests for identification of microbiomes and bring new and cost-effective commercial applications to market. That was supported by scale-up and industrial validation of the production of microalgae originating from Work Package 3 and fermented food from Work Package 4. And then finally, create a better EU agro-aqua food system that is resilient, resource-efficient, climate-resilient, sustainable, and consumer-centered. That was supported by the professional marketing plan, which includes a market potential analysis for launching new innovative products in the market. And just to shortly summarize the results from the standpoint of exploitation. The first one, a pilot-level upscaling procedure for a production of starch strains to be used in large-scale fermentations, can be applied for scaling up of the fermentation processes to obtain a microbial starter for various scaled-up fermentation processes. And the next one, a bio-waste-based culture medium applicable to cost-efficient, large-scale production of plant growth-promoting microbes. It will be important for the production of plant growth-promoting microbes to be used as biofertilizers in agricultural productions, and specifically it contributes to the EU goal of reducing fertilization and chemicals in agriculture. And then a standard operating procedure of cryopreservation technology for preservation of complex microbiomes from various environments. This protocol can be applied for long-term storage of starter cultures or to be exploited in crop cultivation, microalgae production, and food production. And then the next one, simplex, multiplex, and quantitative real-time PCR access is to trace microbial consortia in field samples. This protocol can be harnessed, for example, for preliminary microbial analysis in various samples which originate from soil, plant, and food supply chains. The next one is scale-up of a food production process utilizing selected plant materials and starter microbes. It can be utilized for the development of fermentation protocols for plant products based on optimized microbe consortia to improve nutritional quality and taste at the end of the end product. And the final one, professional marketing plan for microbial products for agriculture that can be applied in the production of new and cost-effective commercial applications to the market, and especially it goes with products in line with directives in EU which aim to the reduction of nitrogen and phosphorus budget surplus in agricultural fields. Now when there is a number of new innovations coming up, what is the situation in the global market for agricultural microbial products? So over the last decades, the number of competing companies has been growing steadily, and this is verifying that there is growth opportunities in the sector of agricultural microbial products, but also that there is increasing competition coming up in this field. Okay. And in addition to opportunities, there are also specific challenges in the market for agricultural microbial products. And this can be categorized actually in 5 different groups. And the first one is threat of competitors, which is called medium risk, threat of new entrants, which is considered as medium or high risk, the threat of substitute products, which is low or medium risk, and then bargaining power of suppliers, which is considered low risk, and bargaining power of buyers, which is considered medium high risk. One potential sector in the field of agricultural microbial bioproducts is the biofertilizer market. And the application of microbial biomarkers represents a key driver for sustainable crop production, and it fosters a reduction of chemical pesticides and synthetic fertilizers and biofertilizers. In addition to providing an eco-friendly option, they also maintain the soil and crop health with increased efficiency. Actually, it seems that there is one slide missing, but I don't think it matters because we are already very close to the end of the time. The regulatory situation in the agricultural bioproducts is still in the development phase, but certain basic rules apply. And in the first place, in the European Union, European Food Safety Authority, EFSA, assesses the safety of microorganisms which are used in so-called regulatory products before they are authorized for the use in the European market. And these regulatory products cover, for example, feed additives, food enzymes, food flavorings, novel food, and plant protection products, so quite a wide variety of different type of products. And to streamline the regulation process, the concept of qualified presumption of safety, the QPS system, was introduced in 2007 to facilitate the safety assessment of a regulated product which is proposed for market authorization. And basically the concept simplifies the evaluation of applications which are submitted to the Commission by reducing the number of studies which are required for an adequate safety assessment of a microorganism if it has a QPS status. So if a microbial species is included in the QPS list and it will be applied to develop a product intended for either human or animal use within the EFSA context, then there is no need for tolerance studies in target animals, studies on consumer and environmental safety and genotoxicity and subchronic toxicity studies if the organism has the QPS status. And at the moment, the QPS list contains a number of bacteria and yeast species, but filamentous fungi, for example, are excluded. And that's because the often very complex secondary metabolism which they have, and it indicates that it's not possible to extend the QPS status into the group of microorganisms like filamentous fungi. And the QPS list is updated every 6 months, so when and if you need some information from there, then make sure that you are looking at the latest release of QPS. Yeah, that's the final slide. Yes, thank you very much for listening.
Okay, thank you, Vesa. Now, if there are some questions, you have a few minutes.
Just a short one. I really enjoyed your presentation. But the filamentous fungi, I believe the Japanese miso, tempeh, etc., are included, and they are beautiful filamentous fungi.
Yeah, they are actually those ones which are included are such which have a very long history in food production. That's also in Nordic countries. There are some fungi used in for example, fermented milk products. But if you tend to use a same species but a different strain, then it may not be possible to do it via this QPS route, because it's impossible to say if, for example, if it goes to Aspergilli, as you mentioned, the Japanese products, which are very risky in that sense that some strains produce mycotoxins and others not. So it's very, very difficult to convince anyone that even though the species is the same, that there is no difference between those different strains. Yeah.
Thank you. I'm particularly interested in the task, or what you achieved in task 6.2, and could you tell me how did you assess the stability of the cryopreservation of these complex communities, marine, and how successful it was overall? So
yeah, there were revival tests after cryopreservation, but I think we have the experts sitting here who actually did those jobs. So I hand over to Henk to give precise information on this. And maybe Anna-Maria also maybe has something to add to that.
For the cryopreservation, there were two problems. First of all, you have to preserve the mixed community. But for us, the success of the cryopreservation was only if we could regrow the community. So if we could regrow the community in a similar setup with a similar community after growth. And what we found is there were several cryopreservatives that did a good job, but they had a negative effect on regrowing the strains. For example, glycerol and dimeso, they are very big stimulants of certain heterotrophic bacteria. So you create an enormous anoxia, and this was a photosynthetic-driven coastal microbial mat. And so in this case, methanol, or not doing anything, was the best to maintain a community that was closest to the original community. But we could freeze them at minus 150 degrees for, we tested up to 150 days.
Thank you.
Yeah. Yes, thank you very much for listening.